I'm Mark DePristo, CEO of BigHat Biosciences. I'm delighted to be here at the SMI AI and Drug Discovery Conference in March 2021 to tell you about antibody design using synthetic biology and machine learning. The plan for this talk is to first do a high-level introduction to AI/ML methods, talk a little bit about their applications to drug discovery in general, and then do a deeper dive into antibodies and BigHat Biosciences itself. I like to start these talks with a quick overview of my background so everyone can understand where I'm coming from. I've had about 20 years of experience at the intersection of applying technology and biology. My background was computer science math guy, and I went ultimately to the University of Cambridge, got a PhD in biochemistry, spent a number of years at Harvard doing actual experimental biology, got pulled into human genome sequencing at the start of the next generation sequencing revolution and led the 1,000 Genomes Project at the Broad Institute, developing a lot of the methods for analyzing next-gen sequencing. And after a brief stint at a startup in Boston, I moved out to San Francisco and joined Google's AI division, where I founded and ran the genomics team there. And in 2019, I left Google to found now Series A startup called BigHat Biosciences, applying AI technologies to antibody design and engineering. Before we get too far into the application in biology, let's start with an overview of artificial intelligence, machine learning technologies today. So I like to think about machine learning as a field primarily interested in building automated computational methods that can learn from data to perform some task. And within that space, there's really three main pillars. There's supervised learning, unsupervised learning, reinforcement learning. We'll start talking about supervised learning because it's the most common in biology. Supervised learning is really focused on this question of learning to predict some output given labeled training inputs. And an example here would be a slide from pathology that has cells that might be typically developing, some might be cancerous, and you want to have a computer program that can look at that image and annotate which cells have cancer and which do not. And in this scenario, the training data would be, coming from an experienced pathologist who knows which cells are cancer or not. And we have methods that can take lots of example images and which cells are cancerous and not, and learn the pattern that allows them to label new images that don't have labeled data from an expert pathologist. A contrast to supervised methods, which have labeling as a key piece, unsupervised methods really don't have labels. And the goal here is really to uncover patterns in the data from unlabeled data sets. So this is often things that you might think of as visualization with DSNE or PCA for dimensionality reduction, or clustering, finding how many mixtures there are in a say Gaussian, a mixture of Gaussians. These methods are often used in sort of discovery science, and I think of them as very typically used today in, for instance, single-cell RNA sequencing, where you might ask, how many different cell types do there appear to be in the data? Where, of course, we don't know all the different cell types, so we can't really label that. And finally, there's the reinforcement learning arm of machine learning, which is really focused on learning actions from feedback in some environment. And this is, in some sense, the best thought of in areas like AlphaGo, where you're learning to play Go or you're learning to play chess. But these methods are also very useful in biomedicine, where, for instance, you might want to learn to control a surgical robotic arm to make it possible to do surgeries without surgeons. This really, the big question is, why are people so excited about AI and machine learning today? It's an old field. It's been around since the 1950s. And that's because there's been a real recent breakthrough in so-called deep neural networks. You can hear this called deep learning. They're really extensions to some of the earliest machine learning technologies. We invented neural networks. But as you can see here in this illustration, the fundamental thing that makes it deep is that there are many layers in this hidden function. And so the way to think about this is you're building a function f, which is the deep learning model that tries to predict an output y from a bunch of inputs. So here's an example of learning to classify whether there's a cat or a dog in the image by sending them through a very deep network that can learn the patterns that make a cat image a cat and a dog and an image a dog. The real excitement from these methods, which have been in developed for 20, 30 years, really accelerated in the 2010s as really deep networks because of really explosion of data and computer processing broke through what have been major accuracy ceilings. So if you, for instance, look at the performance of predictors on the ImageNet challenge, there's really a discontinuity as deep learning technologies came in. And that ability to suddenly perform at human or superhuman levels on challenges like image recognition have driven the major excitement in bringing these technologies into other fields besides, obviously image processing. As I alluded to on the last slide, the biggest driver of the explosion of deep learning interest over the last decade has been the vast growth in the data sets that we have access to, which have allowed neural networks, which particularly deep neural networks, to overcome this sort of performance limit. And it's useful to understand exactly a little bit that dynamic. And this is based on an original slide from Andrew Ng that really explains it, I think, critically nicely. The way to think about it is on the x-axis, there's some amount of data. You can measure it in any number of ways. On the y-axis is sort of the performance of the task. And when the data sets are really small, traditional machine learning methods, which are generally more constrained than the deep neural networks are, they just are better at making use of limited data because they, in some sense, inject more constraints or priors into the situation. And so they outperform the neural networks. But as you gain more and more data, the traditional methods, which had clearly plateaued in their performance, suddenly there's a shift. The neural networks cross this critical threshold where they have enough data to start matching and then outperforming those traditional methods. And a key piece, which I would say is is my editorial on top of this, is one of the biggest observations in the last few years is that it's not really just the total amount of data, but really is the total amount of the right type of data. So it has to be high quality, it has to be labeled accurately, and it has to be useful for training. And that's an important concept to keep in mind. As we transition into this application of where AI technologies are going to be useful in bio, I wanted to set the stage for just the general question, how do you recognize good AI ML problems in general? And I think about this as coming from the trinity of having the right shape, the right difficulty, and the right data. And the shape is really a question of, is the problem an actual prediction problem or equivalently a supervised or reinforcement learning problem, if that's how you're thinking about it? But I'm going to focus on the prediction or supervised class. So what you really need to look for in a problem is something where there's a variety of inputs that can be measured. Those inputs go into some complex process, which is too hard for you to directly model or understand. But then it magically comes out on these outputs, which you can then measure yourself. And what you're really interested in is building a model for that complex process. And so if for any kind of problem that you're working on, if you can formulate it as some complex transformation from a bunch of inputs to a thing that you can measure that's the output, then maybe you can build an ML model for it. But of course, not all problems that can be formulated as prediction problems are particularly good AI ML problems. And that's because you really need to be in this magical Goldilocks zone of difficulty. It can't be too easy and it can't be too hard. And too is simple, simple methods would have worked. There's probably very good predictors that already exist. And if the problem is too hard, it could be that it's irrelevant that we have advanced AI ML methods and they're simply impossible to solve. This is an example of, for instance, you might have the incorrect inputs. You may not have almost any predictive power from the inputs that you can measure. So the key is to define these problems in this just right Goldilocks zone where You clearly are going to be able to leverage the recent advances in AI/ML to build predictors that are much better than the state-of-the-art predictors today. And this is really where you should focus on finding problems. And then finally, and I want to emphasize the criticalness of data here, is that in order to be a really good problem for modern ML systems, they're going to need a lot of data, as I mentioned previously. So you really want to pick problems where you have access to new technologies to measure the inputs and outputs at scale, right? Because ultimately, in sort of the dark secret and sense of most AML work, is that success is largely determined not by your access to novel algorithms, but by access to quality data. And so you really want to look for problems that are data rich or have the potential to be so. You might have new ways to measure the inputs and the outputs, or you have new metrics that you can include in the inputs and outputs. And those are all good signs for new types of problems. With that introduction to AI/ML methods under our belt, we can start to really ask, where are these innovations occurring in drug development, and where are some of their key applications? And at the top of this figure, you can really see the breakdown of drug development from target discovery through creating the therapeutic modality to preclinical, clinical development, and ultimately landing in the market. And I highlighted a couple of places where I've seen, and many other people see, major opportunities for AI/ML applications. In target discovery, you can really think about this as multi-factor, multimodal target identification or biomarkers. At the bottom of each of these sections, there's a link to a recent review article that covers this area, so I won't go into too many details at this stage. Once you get past the target discovery, you have something in hand that you want to actually affect, that you believe is going to affect the disease state. You want to move on to creating a therapeutic. This can be anything from better screening technologies that'll let you identify, small molecules or proteins that are very likely to be good therapeutic candidates, to things like novel approaches to molecular design and engineering, which incorporate, iterative feedback similar to a superpowered directed evolution approach. And finally, I wanted to highlight that clinical development as well is seeing some major inroads for AI/ML technologies, largely focused around either selecting patients in a more intelligent manner, so you have a better chance of succeeding, to actually designing trials with a very different structure. So you have a more efficient mechanism for uncovering safety and efficacy data. And I should emphasize that all three of those are what I would call direct applications. They directly affect the key steps of drug development, identifying targets, identifying therapeutic agents, and demonstrating safety and efficacy. But there are a lot of indirect applications and improvements throughout the development stack that are, I would say, more general than just drug discovery, but are significantly improving the process itself, from molecular modeling. So this would be where AlphaFold would fall, to the massive increase in computing power provided by GPUs, largely driven by the AI applications, and of course data analysis itself, where we have advanced vision algorithms and things all based on modern technologies in AI/ML that will move the needle in drug development, indirectly. So I'm going to dive into two of those three examples in more detail, try to explain a little bit how more they work. Let's start with the big one of target discovery. How do you identify targets in the genome that are likely to change the disease state of your disease if you were to modify them in some way? And traditionally, this has been a very manual and difficult process. And what's occurred recently is a massive explosion in large-scale data sets, that cover relevant to this space, everything from, human genetics data sets to pathways to large-scale functional analysis, and et cetera. And I think we all appreciate that integrating this information would give us better and more holistic views on which targets are good, but it's extremely difficult to integrate so many different types of data using traditional methods. The AI ML technologies are actually very good at multimodal data integration and are a great area where I see a lot of improvements in AI approaches for discovery. And I wanted to emphasize that there's really two problems that the AI ML systems are able to solve. And they provide, more than just a statement, this is a good target or not. They give you some sense of a probabilistic estimate. But there's two real pieces, right? One is identifying the actual target. This gene is involved in a disease. You can envision that as, giving you an estimate of the probability of that gene being involved in disease, given that there's some target. The other way, and really even potentially more important component, is the therapeutic hypothesis, which is to really show, that there's some evidence that if you change the target in some way, that you'll modify the disease state. And this is a particularly good example when you think about cardiovascular disease and HDL. All those genes are obviously involved in cardiovascular fitness and the chance for MIs, but modifying HDL levels turn out to not actually modify cardiovascular risk, which is an insight that came out of human genetics recently, and AI ML technologies can really help to tease out those sort of causal relationships as well. And then another area that I want to highlight, which is really, near and dear to BigHat Biosciences, where I'm the CEO, is doing better molecular design with AI. And this is really to go from the traditional approach, which is sort of random experiment-driven screening approach, where the figure of merit there is really to find larger libraries. But with AI ML, you can really do a data-driven design. So you're replacing this experimental component where you're diversifying your library with something that's using machine learning to guide the mutational process, which then becomes a specific set of mutations and really it goes through a set of kind of more discrete assays with the goal of producing better molecules more reliably using AI ML technologies. And unlike the traditional approach where the goal is to have, very, very diverse and large libraries, you could think of the AML approach as being really focused on how many cycles of exploration you can do as you guide your search toward the best molecules. So with that, let's talk about the area of BigHat Biosciences, which is really this deeper application of AI ML technologies to molecular design. I'll start with an introduction to antibodies in general, and then talk increasingly about where BigHat fits in this. So BigHat really is focused on the design of antibody therapeutics, which is a huge, multi-$100 billion a year market. It represents most of the top selling drugs last year, and it's been transformative for the treatment of diseases. As you can see from 1939, there was really only 3 ways of treating cancer, most of which were not super effective. So we've luckily benefited from the chemical and now biologics revolution in cancer. But that revolution, which was spearheaded by monoclonal antibodies, has seen really an explosion of these so-called next generation antibody formats, which have all been starting to be approved in the last 6 or 7 years. These are invented to really overcome the limitations of these monoclonal antibody formats. There's all sorts of undruggable targets for them. There's inaccessible mechanisms of action. The example is MABs are only good at binding one thing versus BiTEs, for instance, bind 2 different molecules and glue them together. So these so-called next generation formats have been, incredibly influential and important for patients. But as we've started to create these next gen antibodies, we've realized that designing next gen antibodies is substantially more challenging than their monoclonal parents. That's because the biophysical properties of many of these next generation antibodies are just worse on average than what the monoclonals are having. And these poor biophysical properties, like state instability or insolubility or poor yield, turn into low potency or safety or other types of manufacturing problems. And that's in large part because many of these designs are really more human guided than a mAb, which is more like a natural product. And what we've realized is that this design challenge to produce ultra high quality biophysics in these design molecules is a huge issue. And it's really limiting our ability to create these molecules and bring them to patients today. The difficulties of designing next generation antibodies really stem from limitations of our existing technologies. On one hand, we have very sophisticated screening technologies that are able to take a pool of molecules and pull out molecules that have specific high quality individual property, but it is very difficult to screen for many properties simultaneously. And when you pull out your sort of favorite molecule from that pool, and it maybe has got high affinity but not quite the stability or solubility you want, you're forced into expert-driven mutagenesis, which is very difficult. It's both slow and unreliable, and that leads to this difficulty of designing next generation antibodies. And we have a vision here at BigHat of supplementing those approaches with two key types of innovations. One is a high-speed wet lab that can build and test antibodies very quickly at very low latency, and coupled that with active learning technologies that can guide the design for antibodies that are really high quality, not just in one single property, but all the properties that we care about and can measure in the lab, all simultaneously. We're building two key technology stacks here at BigHat that let us create better antibodies faster. One is really design modules with AI and a build test wet lab that collectively allow us to iteratively design molecules that have better properties. And so really to understand this, imagine you're starting with an antibody sequence that has, properties that need to be improved. It's insoluble, it's unstable. The way this process works is we generate a diversity of designs from the AI modules that introduce mutations throughout the antibody that are likely to improve those properties. Those go to the BigHat work cell for rapid, production, characterization, producing, data on all of these biophysical and other properties of the molecule, which then cycle back into the AI ML models, which now improve their prediction, and a new round of better designs can be made. And the critical thing to appreciate about this process is it's an iterative learning system. It gets better with use, not only on individual campaigns as you're optimizing, say, one antibody sequence, but it gets better over time as it learns to the real patterns that are shared across molecules. So let's take a little bit of a dive into the synthetic biology technologies that BigHat uses to power its work cells. We define the work cell at BigHat as really going from in silico designs in our cloud-based design suite to back full antibody characterization data in the cloud again, and it flows through BigHat's work cell in our lab in San Carlos, where we're doing DNA synthesis, cell-free protein synthesis, and a variety of gold standard microplate-based assays that collectively allow us to go from about 5 days from an in silico design to full characterization of around 100 antibodies. And that technology is rapidly improving, with a goal of achieving a kind of 2-day or less turnaround in the near future. And of course, that wet lab is coupled to sophisticated AI ML technologies for solving really two fundamental problems that we encounter while doing design. One is we build assay models. These are predicting the properties of mutations to antibody sequences, and allows us to enrich our designs for high quality sequences for every property that we measure on our platform. So an example is, this is a model that we have predicting the effect of stability of a variety of different mutations. The predictors are very good. These are R squareds at like 0.8, 0.9 levels, but they're not perfect. And it gives you a sense of why we have an experimental lab, is because these predictions, though good and certainly able to enrich your designs for higher quality molecules, are not going to be good enough in a near future to just do predictions alone. And so We also have a continuous need at BigHat to pick high quality sequences to actually make on our lab, which has a capacity of hundreds of antibodies every week. So the question is, which of all the sequences that we could make should we make? We want to choose good designs because they might actually identify sequences that are specifically good. So this is a called exploitation challenge versus trying to make novel designs that will improve the model itself. And we have a variety of novel active learning methods here at BigHat to improve the rate at which we learn from smart data. And this is some example data on actually an expression model that we are working on with the National Institute of Science and Technology. And these types of models are rapidly improving within BigHat, and we see a huge, very exciting future for this type of work. So with that said, let me dive into a real world example of BigHat applying its technologies to design an antibody. And in this case, we're going to talk about creating a bispecific antibody against the SARS-CoV-2 virus. And we do this in a very BigHat way. We're actually starting with not discovery of our own SARS-CoV-2 binders, but we actually went out and sourced every VHH and scFv sequence we could find that would bind to SARS-CoV-1 or MERS in the literature. We brought these into the lab and characterized their ability to bind SARS-CoV-2. For the most promising candidate starts, we ran optimization campaigns of all those individual antibody heads to produce high quality binders against SARS-CoV-2. And I'll just talk about an example trajectory there. So, as I said, we start with a sequence of an antibody. We then diversify, introducing mutations to that and characterize them in our lab. And in the first round of our optimization, we identified mutations that improve the binding affinity to the spike protein, roughly 100-fold. We realized, though, as in the middle of this campaign, that actually a more natural optimization target wouldn't be just affinity to the spike, but actually the ability to outcompete the ACE2 spike binding interaction. And so we switched in the middle of the assay in the optimization campaign to that assay and realized another roughly 20x or 30x improvement to this specific head. We had then did this for multiple different heads of the antibodies. We took what we thought would be the most promising that would not be competing for the same epitope, and we optimized the linker between these two VHH heads to make a highly potent molecule, which we then sent into BSL-3 lab for viral neutralization testing and demonstrate that this bispecific antibody we've created is extremely potent, relative to, for instance, the Regeneron or Lilly cocktails, which are on the market now with potencies in the tens of nanomolars per microliter, and ours is down at kind of 2. Thank you for taking the time to listen to the story about AI and applications in drug development and antibody engineering. I want to leave you with 4 key takeaways from this talk. One is that AI/ML is really here to stay. It's a serious technology and it's improving rapidly, and it has an enormous number of significant contributions to make throughout the drug development cycle. One of the major areas for AI/ML in drug development is really better molecular design and engineering, to make the therapeutic agents that we use to modify our targets. Biologics, particularly next generation antibodies, really require more advanced molecular engineering technologies, which is the commercial focus of my company, BigHat Biosciences, which is integrating synthetic biology and AI/ML techniques to improve how we design and engineer antibodies. Hope that this talk has convinced you that this is a really exciting area, that there's lots of promise here, and anyone who is interested in learning more about the area, about antibody design, about machine learning applied to antibodies, or BigHat Biosciences, please don't hesitate to reach out. Thank you very much.